Good morning. For this week, we are going to focus on the cells of the nervous system and how they communicate. Um, this lecture is going to focus on the cells of the nervous system, and the next video is going to focus on part of neuronal communication or the nerve impulse. So I'm going to get started by sharing my screen. So the nervous system is made up of two types of cells. We have neurons and glial cells. The human brain contains approximately 100 billion individual neurons, but this number is going to vary from person to person. So that's a general or average amount. But all of our behavior depends upon the communication between neurons. So neurons are specialized cells in the nervous system that receive information. They conduct electrical impulses or transmit information to neighboring neurons through the transmission of electrochemical signals. So the next video is going to focus on the electrical portion of the electrochemical message, and then the following week we'll hop into the chemical portion of those signals. But neurons convey sensory information to the brain. So our sense systems are what allows us to stay in direct contact with the environment. So we have sensory neurons that will send information from those receptors or neurons to the brain. And there are other neurons that are going to carry out operations that are involved in thought, feeling, and action. So they transmit commands out into the body. So from the world into the nervous system, and the nervous system into the body, and then allows us to act on our environment. Now, neurons have many sizes and shapes. Here's just an example of our prototypical neuron, and we're going to talk about the features of a neuron in a bit. But again, they have different sizes and shapes. And the size and the shape is going to be related to their function. So a sensory neuron is going to look different than a motor neuron or an interneuron. And even neurons that are in our visual system are going to look different than the neurons that are in our hippocampus, for instance, which is a structure that plays a role in memory. So their shape and their size is related to their function or what role they play in processing information. Really how many connections that they make with other neurons. So what I'd like to do for a moment is just hop out of here, and I have this link. So neuroanatomist, illustrator, Ramon Cajal made these illustrations in the late 1800s. And he was the scientist that proposed that neurons don't grow into or make direct contact with one another. And the microscopic technology wasn't what it is today. So to have that theory given the technology was revolutionary, and some disagreed with him at the time. We now know that neurons do not actually touch one another. We'll talk about the gap that separates them and its function next week. But I wanted to show you these drawings. There was an exhibit in the Gray Gallery at NYU where they have a collection of his drawings so that you can see different systems or connections for these cells in the nervous system. So these are several receptors, multiple cells that are making connections or communicating with other cells. So here's more of a system view. Here are some other cells in the nervous system. See how they have different shapes and sizes. Here is a section of the brain, I think. Oh, this is glial cells in the spinal cord. We'll get to glial cells in a moment. This is a Purkinje cell. Yes, Purkinje cells are visual cells, and some are in the cerebellum, and we'll talk about the function of that part of the brain in chapter 4. But as you can see, this cell has a lot of branching fibers here, and that this looks quite different than the cell that we opened with. So that looks quite different from this cell here, in that there's just more of these fibers, which we call dendrites, but we'll get to that in a moment. Okay, so neurons communicate with other neurons using electrochemical messages. Glial cells, on the other hand, primarily communicate using chemical signals, but some communicate using electrical signals as well. There is much less known about glial cells than neurons. And scientists are discovering new classes, subtypes of these classes of glial cells. So firstly, they outnumber neurons about 10 to 1. There's actually 10 to 50 glial cells for every one neuron. They're thought to be, generally speaking, support cells for the neurons. But there's recent evidence that glial cells can communicate and moderate the effects of neuronal communication. Those cells are the astrocytes, which we'll talk more about in a moment. Okay, so there are 4 classes of glial cells. There's the microglial, the radioglial, astroglial, and oligodendrocytes and Schwann cells are cells that make myelin. And we'll talk about what the myelin is and what it's for when we're talking about the external structure of a neuron. But briefly, myelin is just a fatty substance that coats a part of a neuron that aids in its sending of the message, or the integrity of the nerve impulse, the signal being sent. Now, oligodendrocytes and Schwann cells both make myelin. The oligodendrocytes make myelin in the central nervous system, so the brain and the spinal cord, whereas the Schwann cells make the myelin in the peripheral nervous system. The subtype of astroglial, called astrocytes, what they do, they have multiple functions. So they actually provide a physical scaffolding for the neurons. Without that scaffolding, neurons would just float freely in extracellular fluid, but the astroglial give them support and stability. They aid in the holding of the connections between neurons. They moderate signals from neurons. It's thought that they help to synchronize the activity of neurons. So messages communicate in bursts or in waves. They provide rhythms of communication. And we'll talk later on in the semester what happens when areas of the brain that have synchronous rhythmic patterns versus asynchronous patterns. But the astroglial help to regulate this through their signals. They also help form the blood-brain barrier, which we'll talk about in a moment. Okay, so they're quite the support structure for the neurons. The radial glial we'll talk more about in the chapter on development, but they guide the axons to targets during development. So when we are developing, when we're born, we have more neurons than we need. And what happens is when they communicate to and follow pathways of communication repeatedly, those connections strengthen, the others prune away. Prior to that in development, certain areas of the brain send chemical messages that attract the growth of neurons to reach those particular targets, and the radial glial help guide the axons to their targets. Then there's microglial, which have immune system responsibilities. So they play a role in cell death and synapse formation. So the systems of communications or pathways of communication between neurons, and synapse elimination. So at this point in your education in psychology, you've probably heard the term use it or lose it, right? So when there are networks that were formed that are no longer being used, those synapses get eliminated. So the axons of neurons can form new synapses. Microglial help in this process. We're going to talk about cell death more, especially when we're in the development section, but cells can die for multiple reasons. It's important to know that aside from a few areas in the brain, neurons do not regenerate themselves. If there is a virus that gets in the brain and kills the neurons or damages the neurons, the microglial clean up the parts of the cells. If neurons don't receive transmission or messages from other neurons, they will potentially die. Microglial cleans up those neurons. Okay, speaking about viruses entering the brain, that's not a great thing, right? As I was just saying, if a virus is able to get into the central nervous system and damage those neurons, they are not going to regrow. So to guard against the possibility of that happening, we have what's called the blood-brain barrier. And it's a mechanism that surrounds the brain and blocks most chemicals from entering. So it blocks viruses and bacteria from entering the brain, but it also blocks healthy chemicals chemicals that we need, even the brain's primary nutrient of glucose, from readily crossing this barrier. So again, like I said, neurons in the brain generally do not regenerate, so it's vitally important for the blood-brain barrier to block incoming viruses, bacteria, or other harmful materials from entering. The blood-brain barrier is made up of cells, blood cells that are packed very tightly together. More so than we see them in other areas of the body. And again, this blocks chemicals from entering the brain, but it also blocks the primary nutrient from entering the brain and other chemicals that we need. So in order for those chemicals to cross the blood-brain barrier, we have what's called an active transport system. And so that system takes resources, it takes energy, right? So you have this system that will transport glucose, for instance, into the brain that requires energy to move that chemical across the blood-brain barrier. So that should just tell you something about how important there is. There's a metabolic cost to getting the brain's primary nutrient into the brain. But the cost would be too high to not have a structure like this, because again, neurons don't readily regenerate. Some do. Neurons in our olfactory system, right? The system, our sense system of smell, and in our hippocampus, which again plays a role in memory and the creation of new memories. So going back to songbirds, right, if we look at the hippocampus of songbirds, they also grow new neurons in their hippocampus, often seasonally. And another interesting thing to note is that in illnesses such as Alzheimer's and related illnesses, the gap in the cells that make up the blood-brain barrier have gotten larger, right? And again, that's not good. It's not going to block all of the possible infections in the body. Okay, so let's talk about neurons. Okay. So as I said before, neurons are going to vary in size and shape and their function. And their function is related to where in the brain they are. And that's going to be related to what size they are, what shape they are, how many connections they make, so on and so forth. So the shape of a neuron determines its connection with other neurons and contribution to the nervous system, right? So is it responsible for something having to do with learning and memory, or how we get input from our environment, or how we send motor commands so that we can explore our environment? Again, the function is closely related to the shape. Right, so here's another figure that's really displaying what I showed you from those Gray's gallery images, which I think is much nicer to look at than this particular figure. But you can see the difference between Purkinje cells and bipolar cell in the visual system. I think these are receptors for touch. I'll talk more about this. Okay, so now we're going to talk about the internal features of the neurons and the external features of neurons. And so neurons are similar to other cells in the body in that neurons contain the following structures. So they have a membrane, a nucleus, mitochondria, ribosomes, and the endoplasmic reticulation. And I've defined all of these in the coming slides. Right? So the membrane is a structure that separates the inside of the cell from the outside environment. So it's composed of 2 layers of fat molecules, and it has what are called protein channels. And these protein channels let a few charged ions to cross the membrane, but most chemicals are not allowed to move through that membrane. This is going to become important when we talk about how the neuron communicates. So there are channels in the membrane. Some particles are allowed to flow freely in and out of that membrane through channels. Some channels are going to be what's called voltage-gated protein channels. We'll talk about that more later. But again, there's channels that allow ions to flow in and out of the membrane. Neurons have a nucleus, which is a structure that contains the chromosomes. The mitochondria is the structure that performs metabolic activities and provides the cell with energy. Ribosomes is the site at which the cell synthesizes new protein molecules. So we're going to talk about that next week in relation to neurotransmitters, or the chemical portion of neuronal communication. And the endoplasmic reticulum is a network of thin tubes that transport these synthesized proteins to their location. And again, we're going to talk about that when we talk about the chemical portion of neuronal communication, which again is an electrochemical process. Again, those are the internal features of the neuron. Now we're going to talk about the external features of a neuron, which really distinguish the neurons from other cells. And when we talk about this, we are talking about it in terms of like your prototypical neuron, which looks like that image that is in the previous slides. Again, neurons vary in their shapes and their sizes. Some of them are missing some of these features, but we're going to talk about just the prototypical neuron. Okay, so I have the components listed on this page, and we're going to talk about each one. I think it's easier to talk about it when we're looking at a visual depiction of a neuron. Okay, so neurons have dendrites. Dendrites are these branching fibers that receive incoming information from other neurons. So their surface is lined with specialized, what are called synaptic receptors. And again, that's the site of the receiving the incoming information. So these dendrites, these branching fibers, can also grow what are called dendritic spines. What that does is it grows the area of receiving information and can make connections with more neurons. This part of the neuron is called the soma, or the body of the cell. The body of the cell contains the nucleus, the ribosomes, the mitochondria, and other structures that are found in most cells. So the genetic material and the energetic work of the cell and synthesizing of proteins is happening in the cell body. Okay. Now, if you see this, I should have been using this neuron here to talk about the structures of the cell. Sorry about that. This is also a cell body with dendrites here. But this is actually the presynaptic neuron. This is the one receiving a message from this cell. So it's called the postsynaptic neuron. But they're both going to have the same structures. You can just see the complete version here. So again, dendrites, they can have branches that grow off of them called dendritic spines. The cell body, and this branching fiber coming off of the cell body is called our axon. And the axon is the portion of the neuron where the electrical message is going to be sent down. So it's a long, thin fiber, which is the information sending part of the neuron. So again, it sends the electrical portion of the communication to a receiving or a postsynaptic neuron. This portion of the neuron right here is called the axon hillock, which is approximately where the nerve impulse is generated. The electrical portion of a message is generated. Now, see these oblong shapes here? These are the myelin. This is the myelin sheaths. And it's insulation covering some vertebrate axons. Not all neurons have axons, and not all axons are coated with myelin. It depends on how long the axon is, which is also informed by where in the nervous system they are. So if a message has to go from your motor cortex to your pinky toe, right, that is going to be, there are going to be axons there that are coated with myelin. Now, there are gaps in this fatty substance, as you can see here. Those gaps are called nodes of Ranvier. And we'll talk about this more when we talk about the electrical portion of transmission, but it regenerates at these gaps in this fatty substance. So we have an axon, which sends the information. It is covered with myelin, fatty substance that helps message transmission, keeps it from degrading. These gaps in the myelin are nodes of Ranvier. That's the site of regeneration of the message, of the electrical message. With an illness like multiple sclerosis, the immune system attacks the myelin sheath. It attacks this coating. And so the electrical message may degrade before it reaches the postsynaptic neuron or its target neuron. And if this neuron can no longer send its message, it will die. And then the microglial will swoop up and clean it. If this neuron is in the central nervous system, the myelin was made by oligodendrocytes. If this neuron is part of the peripheral nervous system, it was made by a Schwann cell. Okay, so let's go down here. The end of an axon, you will see what are these little bulbs here. This is called the terminal buttons, or sometimes just buttons, or sometimes presynaptic terminal. And these buttons contain chemical messengers. Again, the neuron communicates electrochemically, and here is where some chemicals are housed, and chemicals are released from here, and they cross over this small gap. There's small gaps here called a synapse, and make contact with the dendrites of the postsynaptic neuron. Okay, so neurons may have any number of dendrites, but they're limited to one axon. The axon may or may not have branches. It depends on what type of neuron it is. Now, this figure is also a little misleading. There are going to be multiple neurons that are synapsing with other neurons. They exist within networks, and there are multiple cells within the network. So we're trying to take the most simplistic view of neuronal communication at this early stage, but just know that there are networks of neurons. There are multiple neurons communicating with other neurons, and they house different types of neurotransmitters, which are the chemical portions of the communication. Okay, so that concludes our discussion of the cells in the nervous system.